Good morning, students, and welcome to NPTEL live session for the course Introduction to Developmental Biology. Hi, Ajay, and to all other students who will be watching this video. So, the NPTEL requested us for a extra session for all those students who wanted to have revisions and join in this session. So, what I have accumulated in this session is few questions from and try to cover the whole course. So we had 12 different weeks of lectures where Professor Subramaniam is teaching us about the whole developmental biology and including some questions from each of those modules. I have made a presentation today and we'll be solving one by one, and it will be a good revision for all of us to go through each of these concepts one by one in those questions. So today's session we'll solve some 10 to 11 questions corresponding to each of the module that we have faced in the course Introduction to Developmental Biology. And as your exams are approaching, so I wish this is a good time for you guys to revision and focus, sit down, watch your lectures, and be prepared for the exam. And I know that you all guys will do very good. Okay, we will start at 11:05, 5 minutes past 11, and I'll keep admitting students who are requesting to join this call. Ajay, you have raised a hand, you can speak. Okay, if you have any concepts that you didn't understand, and I happen to remember those concepts, then I'll surely help you. Okay, I'll try to, but if you have attended most of my presentations, and since it is a 2 hour long session, I could prepare 10 to 11 questions at most, and I have one slide each for the questions as well as the explanation, and I hope it will be a good revision for all of the students who will be watching this video. Okay, we'll start presenting. Let me share my screen. We'll start the session. Okay, here you are with the final live TA session for the course Introduction to Developmental Biology, and this is week 13 or bonus week, where we'll be dealing with all sorts of questions from different modules that have been taught in this course. And again, I welcome you all to this live TA session. And the course instructor is Professor Subramaniam, and my name is Ashutosh. I'm a PhD scholar at IIT Kanpur. So the similar format we'll be following here as we had done in the past problem solving sessions. I'll pose few questions, multiple choice questions, similar to what you faced in your assignments, but today it will be from different modules that professor have covered and we have also read, and we'll try to find out the correct answer. So I'll give some time to the students to answer what they think is the correct answer. Then I'll reveal what I have selected as the correct answer, and then we'll understand a bit concept about why the correct answer is the right answer. Okay, with that much of introduction, let's start with the first question. Why did Conklin choose tunicate embryo for fate mapping? Why did Conklin choose tunicate embryo for fate mapping? Option A, tunicates were readily available where Conklin lived. Option B, tunicate embryos have large and pigmented blastomeres. Option C, dyes readily penetrated the surface of tunicate blastomeres. Option D, cleavage pattern is invariant during tunicate embryogenesis. So these are the four options. Now students, you have to tell me what was the reason behind Conklin choosing tunicates for his experiments. If you remember in the first few modules where we talked about embryology, there we encountered different scientists laying down the path for embryology and developmental biology. So Conklin was one of the prominent uh, researchers, scientists, and he established very basic theories, fundamental theories using tunicates. So what is the advantage or what is the reason tunicate was chosen by Conklin? Let's take a minute and gather some answers. I see Ajay has answered already. Option 1 and option 2. Okay. I'll reveal the answer in 1 or 2 minutes. Okay, I think it's time I reveal the correct answer. Why did Conklin choose tunicate embryo for fate mapping? The first correct answer is option 2. Tunicate embryos have large and pigmented blastomeres. And the next correct option is cleavage pattern is invariant during, during tunicate embryogenesis. So these are the two correct options. Ajay has answered one correctly, and tunicates were readily available where Conklin lived. This is what he's saying. But I think for the fate mapping experiment, cleavage plane was invariant in the tunicate embryo, and the blastomeres were pigmented, so you could follow the blastomeres. So Conklin could easily track down the blastomere, which blastomere ended up where. So we will just have a brief look at how the tunicate embryo looks like. So in the zygote form and after it reaches the 8 cell stage, so the tunicate embryo can be divided into animal pole and vegetal pole, and these blastomeres have pigment, and the cleavage plane is invariant. By invariant I mean that the cleavage, the development is autonomous. So we know that cleavage will only happen in a certain direction, leading to the formation of different blastomeres, and each blastomere is destined to form its structures. So in embryonic development, early stages involve series of cell divisions that give rise to various type of cells, eventually from the tissues and organs of the organism. The term invariant cleavage pattern implies that divisions are highly regulated and follow a set pattern. So this is what invariant cleavage pattern means. It is predetermined and it is always a set pattern. It is not random. That makes the tunicate embryos, like we already know that A4.2 will only form ectoderm. So even if you separate A4.2, so this was autonomous specification experiment done on tunicate embryo. If you separate ectoderm, if you separate A4.2, it will form ectoderm. So it is destined, predestined because of the cleavage plane and division of the cytoplasmic determinants. So it is autonomously developing and its blastomeres are pigmented, so it's easy to follow for fate mapping. So Conklin could find out that A4.2 will only form ectoderm, whereas A4.1 will form endoderm, but in presence of A4.2 and A4 and notochord. So here we see this region forms the notochord. So in this method, tunicate embryo helped to lay down important concepts of autonomous specification, and it was used by Conklin. Okay, moving on to the second question. Choo- yes, Ajay, I mentioned that pigmented blastomeres and invariant cleavage plane was the correct answer. Yeah, yeah, pigmented blastomeres. Yes, here is the question again. Why did Conklin choose tunicate embryo for fate mapping? So here we see, okay, yeah, yeah, I agree with you, but I actually have less idea where Conklin lived and if tunicate was abandoned. So was it like where Conklin lived, tunicates were abundant? That that made it a good model organism. Yeah, yeah, yeah. Because the that it was transparent. Those those embryos were transparent and it could be seen through. Yeah, yeah, yeah. I understood. But this was the answer that was accepted. Yes. No, no. See, these are the given answers. So I did not made these questions up. So these are from previous assignments and practice sessions. So I have picked certain questions which I found that these are relevant. So these answers are mentioned or accepted in the previous assignments. So they can be correct, they can be wrong. So they are marked correct. But what you are saying, I find if I find it correct, then it's okay because in the final exam, it's your judgment what you will be answering in these questions. And I don't quite know whether you will have the similar kind of pattern because Professor Subramaniam's assignments they have multiple answers which are correct. Okay. Then option B would be the best answer. Tunicate embryos have large and pigmented blastomeres. It was found out later. Yes, yes, yes. I believe you. Okay. Okay. Let's move. Yes, yes, yes. Okay. Let's move on to the next question. Choose the correct statement. Poly A tail is cleaved from the nascent transcript of RNA before translation initiation. Option B. Phosphorylation of residues at C-terminal domain of RNA polymerase II is a key regulator of transcription elongation process. Option C. Electrophoretic mobility shift assay is to determine pair of proteins interacting with each other. Option D, transcription factor act in a modular fashion, enhancers act combinatorially. Okay, so you have to tell me which is the correct statement among these four. Let's take a minute to collect some answers for all the students uh, to make sure that we are on the same page. All the students will be watching this video. Please think about the answer. Which of the following statements is the correct statement? So this is a question about DNA transcription, translation, and transcription factors. Okay, I have seen Ajay answered option 4. Good. Okay, let's see. Let's wait for some more time, then I'll reveal the correct answer. Okay, it's time to reveal the correct answer. The correct answer to this question, which of these is a correct statement, is option number 2. Phosphorylations of residues at C-terminal domain of RNA polymerase is a key regulator of the transcription elongation process. So this is the correct answer. I'm sorry Ajay, but option 4 is not correct. Option 4, it would have been correct if the terms were reversed. Enhancers are the one which act in a modular fashion. If you remember, modularity in enhancers, and transcription factors are the one which act combinatorially. Means there can be. Yes, yes, yes. So C-terminal tail of RNA polymerase. So that's what I'm coming to. There is a C-terminal domain of the. Yeah, yeah. Okay, no issues. Pol2 is the polymerase, is a major polymerase which acts in transcription of genes. So Pol2 has a C-terminal domain. Phosphorylation of the CTD or C-terminal domain serves as binding platforms for various transcription factors, co-activator, and RNA processing factors. So not only elongation, but phosphorylation at different residues in the Pol2 C-terminal domain can attract various transcription factor, co-activator, and RNA processing factor also. These factors are essential for elongation and processing of the nascent RNA molecule. So if you remember elongation process, side by side there is RNA processing going on, because the immediately after RNA is made, RNA is capped, associated with 5' cap, and it is processed. So side by side, whenever gene transcription is going on, RNA is getting produced. We also have 5' cap attaching to the RNA. The phosphorylation state of C terminal domain affects activity and stability of Pol2 during transcription elongation. So non-phosphorylated Pol2 we can only sit on the promoter enhancer. So what it will do? It will sit on the promoter. But to start elongation or to elongate or to move forward, the phosphorylation tails need to be the tails need to be phosphorylated. So hyperphosphorylation of C-terminal domain is associated with transition from promoter clearance to productive elongation. So this step is called promoter clearance, in which Pol2 can clear from the promoter and start elongation or start transcribing the whole of the coding strand, allowing Pol2 to transcribe the gene effectively. So that is why we have uh, phosphorylations in the Pol2 playing a major role in elongation step, C-terminal domain or C-terminal tail. Cool. Okay, moving on to the third question. The control of gene expression at the level of translation can occur by. The control of gene expression at the level of translation can occur by option A, differential mRNA longevity, option B, alternative RNA splicing, option C, selective inhibition of mRNA translation, option D, ribosomal selectivity. Now, which of these can act as a mechanism for control of gene expression at the level of translation? Let's take a minute to collect some answers. I can already see Ajay has written his answer. Option 3 and 4. In a minute, I'll reveal the correct answer. Okay, that's enough time, and let me reveal what is the correct answer. The correct answer to this question, option 1, differential mRNA longevity. So this can also be a method of control of gene expression at the level of translation, because the amount of time an mRNA stays active in the cytoplasm determines its longevity and also the number of proteins and amount of proteins it can code. So there are certain mRNAs which get degraded very fast, and there are certain mRNAs which persist for longer time. So differential mRNA longevity is one answer. Option C, selective inhibition of mRNA translation. So selective inhibition of mRNA translation, certain mRNA translation can be selectively inhibited, and the best example we encountered are microRNAs. MicroRNAs are certain non-coding RNAs which can inhibit the translation of other mRNAs, their target mRNAs. And option 4, ribosomal selectivity, is also an answer. So we have three options correct: differential mRNA longevity, selective inhibition of mRNA translation, and ribosomal selectivity. And option B is not control of gene expression at level of translation, because RNA splicing is a post-transcriptional event. So after transcription, RNA splicing would happen, and alternate RNA splicing is a post-transcriptional event, but not at the level of translation where control of gene expression happens. So if we see this schematic on our left, we have different regulatory labels: transcriptional control and post-transcriptional control. Transcriptional control acts on the accessibility of DNA and the binding of transcription factor, recruitment of Pol2 and everything. So we have chromatin regulation, transcription factor binding, transcription, RNA processing. So all this comes under transcriptional control. Post-transcriptional control or post-transcription, RNA export, RNA localization, RNA turnover. Turnover means how many RNA are degraded and produced per unit time. RNA translation, protein modifications, and turnover. So all these come under post-transcriptional control or translational control. So RNA processing, which is one form of alternative splicing, will come under transcriptional control. So we have three correct options: RNA longevity, differential mRNA longevity, selective inhibition of mRNA translation, and ribosomal selectivity. I hope this is clear. Quickly moving on. Yes, yes, Ajay. Yes, yes, yes. So as I mentioned, let me give you an example. You know the histone proteins that we have. the nucleosomal histone proteins. So the histone RNAs have a special kind of modification. They lack poly-A tail. They lack poly-A tail at all. So what happens? Histone mRNA are very short-lived. They'll be degraded very fast because histone we do not need histone transcription all the time or translation all the time. Only when the cell is replicating, only then we need more histone proteins to be produced. So those mRNA are removed or degraded very quickly. They lack poly-A tail. So this is one of the modes in which longevity of mRNA is modulated. Okay, moving on to the next question. Developmental potential of a blastomere is greater than what it develops into as a part of normal embryo. This conclusion is consistent with the concept of dash. So here we have some concepts from developmental biology. So I'll read the statement again. Developmental potential of a blastomere is greater than what it develops into as a part of normal embryo. Option A, autonomous specification. Option B, genomic equivalence and differential gene expression. Option C, reversibility of cell fate during the stage of specification. And option D, mosaic development. So these are the four options. Let's wait and collect some answers. So the developmental of the blastomere, developmental potential, is greater than any part of a normal embryo. Now what concept can we attribute this conclusion to? Yeah, I will reveal the correct answer soon. So these questions were the ones which I found very difficult. That's why I have included here in the bonus session. So you might find many answers to be controversial. So these are the correct answers that were accepted in the previous assignments and practice sessions. Okay, let me reveal the correct answer to this question. The correct answer is option B, genomic equivalence and differential gene expression. So this is the correct answer. Genomic equivalence says that all the parts of the body share the same genetic composition. Genomic equivalence, the genome is same in all the organs, all the cells and every part of the body. So what is the genome of a blastomere is same as that what is the genome in any part of a normal embryo. Let's say a hand or a intestine or a mesoderm or an ectoderm. So everything is same in the blastomere as well as the part of the embryo. And differential gene expression, it says that with the same genomic equivalence, with the same genome, differential gene expression can lead to formation of different structures or different cell identities, differentiation of the blastomere in this case. So this is kind of the concept it is put. If there would have been an option of regulative development, then that would have been the best fit to this answer. But for these options, option B is the best fit to the answer. Ajay, you want to say something? Autonomous specification you have marked as the correct answer. So in a way, if you see autonomous specification, it also autonomous specification says without that the fate a cell or an embryo or any part of embryo will develop is not determined by its neighbors. It can form itself without any help. So that as we know, many cases autonomous development or autonomous specification does not come into picture. But if we compare, I think genomic. Yes, yes, it can change. Yeah, no doubt, no doubt. But does it explain the potential difference between blastomere and a normal embryo? I know all the options are really confusing, actually. Yes, yes. Hmm. But if you, for any part of the embryo, it might not be autonomous development actually. For a blastomere, autonomous specification is uh, is working because even if you separate a blastomere, when we implant inner cell mass to generate chimeras, or if we saw experiments where in the blastomeric stage, in early stages, uh, each cell of the blastomere has autonomous potential to form the whole organism. But if we move into a differentiated part of a normal embryo, let's say a limb or an eye or a brain, then it needs conditional specification, or it needs its environment to tell it, or the signaling players to tell it what to become. So I think in that part, autonomous specification is missing. That's why it is not the correct answer. Whereas option B is explaining it very well. We have genomic equivalence in blastomere as well as in the normal parts of embryo, let's say head, limb or head or eye or brain. Then we have differential gene expression in the blastomere as comparison to normal any body part of the embryo. That is why we have potential of the blastomere to be greater and the normal embryo to be restricted. I think that is the reason. So here I have mentioned schematic showing the different potentials of the stages of development. In the zygote stage, we have totipotent inner cell mass. Inner cell mass are pluripotent. Embryonic stem cells, iPSCs, are all pluripotent, which are derived from the inner cell mass. Multipotent are adult stem cells, which are found in organs, and unipotent are differentiated cells, which can just renew themselves but cannot form any different lineage. So zygote is totipotent, ICM inner cell mass ESCs are pluripotent, adult stem cells are multipotent, and differentiated cells are unipotent. And this can be explained well with genomic equivalence and differential gene expression. For autonomous specification, as I said, I shared my views with you. For blastomere, autonomous specification is absolutely correct, but for different parts of the embryo in later in the adult life, we need conditional specification. We need the environment to tell the cells what to become. And reversibility of cell fate, um, yeah, in a sense, induced pluripotent stem cells that you were saying. Yeah, that could be also an answer. Option C, sorry. Option C could also be an answer. I agree with you. It won't be mosaic development, but it also not be autonomous specification. Option C could also be an answer. B and C are both equally correct. Okay, let's quickly move on to the next question. Which of the following are true regarding invagination of cells during gastrulation in Drosophila? Which of the following are true regarding invagination of cells during gastrulation? Option A, invagination during Drosophila gastrulation occurs on the dorsal side. Option B, formation of the furrow begins with relocalization of rho and beta catenin. Option C, in Drosophila, the mesoderm is formed from epithelial cells on the ventral side of the embryo. Option D, the apical constriction is responsible for the expression of twist snail and myosin 1 genes. So a technical mistake here, responsible for the expression, not by the expression. So let me just edit this. It will be for. So option A, invagination during Drosophila gastrulation occurs on the dorsal side. Formation of the furrow begins with relocalization of rho and beta catenin. In Drosophila, mesoderm is formed from epithelial cells on the ventral side of the embryo. The apical constriction is responsible for the expression of twist snail and myosin 1 genes. So you have to tell me which is the correct answer, which are the correct answer, which are the true statements regarding invagination of cells during gastrulation. Ajay has answered 1, 2 and 4. Let's wait for a minute to reveal the correct answers. Okay, it's time for me to reveal the correct answer. First correct option is option B. Formation of the furrow begins with relocalization of rho and beta catenin. And the next correct answer is option C. In Drosophila, mesoderm is formed from epithelial cells on the ventral side of the embryo. So these two are the true statements. Apical constriction, the first statement, invagination during Drosophila occurs on the dorsal side. It actually starts from the ventral side. So statement 1 is not correct. Ajay, you want to say something? Yeah. So invagination during gastrulation occurs on the dorsal side. Is not correct. It occurs actually from the ventral side. And the apical constriction is not responsible for the expression of twist snail and myosin, but it is the reverse. So the expression of twist snail and myosin is responsible for the apical constriction. And the mechanism is via relocalization of rho and beta catenin. So if you have a cross-sectional look at the dorsoventral patterning of the Drosophila embryo, we have twist and snail expression on the ventral side. So this is the future ventral side. We have ectoderm or the epithelial cells. So firstly, all of them are ectoderm. All of them are epithelial cells. But some of the epithelial cells start an invagination from the ventral side, which will later form the mesoderm. So this is ectoderm, this is lateral ectoderm, neurogenic ectoderm. So the neural ectoderm is derived from the ectoderm, mesectoderm and the mesoderm. So this part of the organism will form the mesoderm, which is rich in expression with twist and snail genes. And due to expression of twist snail genes, there are two important proteins, rho A and beta catenin. Rho A is a small GTPase protein that regulates actin cytoskeleton. During furrow invagination, rho A plays crucial role in controlling actin dynamics and myosin contractility, which are essential for cell shape changes and tissue invagination. So what does rho A do? Rho A changes the shape of the cells which are going to form the mesoderm, and these can then invaginate inside. Relocalization, rho A is known to become enriched in the apical membrane of the invaginating cells during gastrulation. This apical enrichment of Rho is essential for formation of actin-myosin contractile ring that drives cell shape changes and tissue invagination. So apical and basal side. So if this is the basal side, this is the apical side. So the enrichment of Rho to the apical side is necessary for cell shape changes. Relocalization of Rho to the apical surface is key step initiating these morphological changes. Similarly, we have another player, beta catenin. Beta catenin is a protein which is involved in cell adhesion as well as in Wnt signaling pathway. So during Drosophila gastrulation, furrow invagination, beta catenin is involved in cell adhesion, tissue patterning. It can relocalize to specific cell-cell junctions to facilitate the coordination of tissue movement. So this beta catenin is also relocalizing to the apical surface and specific cell-cell junctions to ensure proper tissue invagination. And here is a schematic showing the ventral patterning, genes which are suppressing other genes leading to leading to the gastrulation movement of the mesoderm. So snail inhibits FGF8, snail inhibits rhomboid, twist activates bagpipe FGF8 receptor. Due to presence of snail and twist, FGF8 receptor is in the future mesoderm, but the FGF8 ligand is excluded out of the mesoderm. This leads to gastrulation movements in the mesoderm. I hope it is clear to the students. Quickly moving on to the next question. List the correct order of events during dorsoventral axis formation in embryogenesis and Drosophila. So there is a sequence of events. You have to list which is the correct sequence of events. List the correct order of events during dorsoventral axis formation in Drosophila. 1. Pipe synthesis is inhibited in dorsal but not ventral follicles. 2. Ester is cleaved into its active form. 3. Follicle cells are induced to form columnar dorsal follicle cells. 4. Cactus is degraded. 5. Gherkin is localized between cell membrane and nucleus. 6. Spatzel is cleaved into its active form. 7. Toll is activated. And 8. Dorsal enters nucleus. So here is the list of events occurring in dorsoventral axis formation on Drosophila. You have to arrange them in the correct order. And your options are 8513627 option B 31574628, option C 53126748, option D 58417 So these are the options and you have to arrange all these events in the order of their occurrence in Drosophila dorsoventral axis pattern. Let's take a minute to collect some answers and later on I'll reveal the correct answer. So if you remember the whole pathway of dorsoventral patterning, how it starts and what are the mediators and what is the final outcome, then you will be able to answer this question. Okay, I see Ajay has mentioned a sequence. Ajay has mentioned option 3. Okay. Now let me reveal the correct answer to all the students. Absolutely correct. Very good, Ajay. You have answered very correctly. And option 3 is the correct answer. 5, 3, 1, 2, 6, 7, 4, 8. So let us arrange them in order. The first event is Gherkin localization. The dorsoventral axis patterning starts with localization of Gherkin mRNA. Gherkin localization between cell membrane and the nucleus. Then the next event is follicle cells are induced to form columnar dorsal follicle cells. So once the Gherkin mRNA is localized, follicle cells near to the Gherkin, Gherkin localization turn into dorsal follicle cells. Then what happens? In those dorsal follicular cells, pipe synthesis is inhibited. Pipe is another protein. Pipe synthesis is inhibited in only dorsal ventral and dorsal follicular cells, but not in the ventral follicle cells. Then we have ester, which is cleaved into its active form. Then we have spatzle, which is cleaved into its active form by ester. We have a series of cascade proteins, serine proteases, which cleave and activate. Then we have toll activation. When toll is activated, cactus is degraded, and cactus is the one which is holding dorsal and not letting it go inside the nucleus. When cactus is degraded, dorsal can enter the nucleus. So this is the correct order of events. So again, I was saying, if you have to remember this diagram, how and what is the events that are happening. So the first event is oocyte nucleus travels to anterior dorsal side of the oocyte, where it localizes Gherkin mRNA. So we know that oocyte nucleus moves and Gherkin mRNA is localized. Gherkin is received by torpedo proteins. So torpedo is a receptor proteins. Torpedo signal causes follicle cells to differentiate to dorsal morphology. So the ones near to the Gherkin become the dorsal follicular cells. And then in these dorsal cell, pipe synthesis is inhibited. Since pipe synthesis is inhibited, ventral follicle cells whereas synthesize pipe, and pipe signals sulfates ventral vitelline proteins such as gastrulation defective. Then these vitelline proteins can bind the gastrulation defective and target the chain of the serine protease pathway. So we have ester first, then comes spatzle, then comes toll. Now toll deactivates cactus and leads to dorsal going inside the nucleus and transcription of dorsal genes. Dorsal protein enters the nucleus and ventralizes the cell. So dorsal will transcribe ventral genes because, and the protein is named dorsal because a mutant in such a gene led to dorsalization of the embryo. So it is named after the mutant. So the dorsal protein codes for ventral genes. Okay, I think it's good enough revision. Let's move on to the next question. Which of the following statements are false with respect to pattern and axis formation in the Drosophila embryo? Which of these following statements are false? Option A, distribution of different gap gene products overlap. Option B, homeotic genes control expression of gap genes, pair rule genes and segment polarity genes. Option C, nanos is a maternal effect gene. Option D, nanos is a gap gene. Ajay, you can mention your answer in the chat box and we'll wait for all students to answer. Okay, I see option 3, 4. Let's wait for some more answers. Okay, let me reveal the correct answer to this question. Which of the following statements are false with respect to patterning and axis formation of the Drosophila embryo? The first correct answer, the first false statement, nanos is a gap gene. This is a false statement. Nanos is not a gap gene. And the second false statement is option B. Homeotic genes control the expression of gap genes, pair rule genes, and segment polarity genes. So this is not the case, whereas it is the other way around. So distribution of different gap gene products overlap is a true statement, and nanos is a maternal effect gene. So if you remember the hierarchy, maternal effect genes are the ones which are the earliest expressing genes, which are laid down in the egg from the mother. Maternal effect genes are the genes which determine anterior posterior axis, and these are the genes which induce gap genes. So nanos, example of maternal effect genes are bicoid and nanos. Bicoid is the head forming or head anterior gene, head demarcating. Posterior gene is the nanos. So these are both antagonistic. These are maternal effect genes laid down in the embryo by the mother, and even before fertilization, they pattern the embryo into anterior and posterior. So the first occurrence are maternal effect genes, then they induce gap genes. Gap genes induce pair rule genes. Pair rule genes induce segment polarity genes. So this is the order. Maternal effect to gap genes to pair rule genes to segment polarity genes. And all these three genes, gap genes, pair rule, and segment polarity, all three of them then induce Hox genes. So Hox genes are the terminal of the hierarchy. That's why this statement is false. The Hox genes or homeotic genes do not control the expression of gap genes, whereas it is the opposite. The expression of gap genes, pair rule genes, and segment polarity controls the Hox genes or homeotic genes. And nanos and bicoid are prime examples of maternal effect gene. They are not a gap gene. Gap gene would be examples would be giant, cripple. These are gap genes. I hope it is clear. Ajay, are you clear? Oh, no, no, no. Bicoid and nanos are maternal effect. You have to. Yes, yes. So we have bicoid and hunchback, caudal and nanos. Which one? Yeah. Yeah. Selector and regulator genes you're asking about. Yeah, yeah. So we have, yeah, we have ultra bithorax and yeah. But definitely homeotic genes cannot. Yeah, yeah, yeah. But definitely homeotic genes cannot control expression of gap genes. Gap genes will only come with in response to the maternal effect, and it is established. So if you see this figure, this is just from Gilbert. So all these gap genes, pair rule, and segment polarity determine the expression band of Hox genes. So wherever we have, yeah, it can be back regulation as well. as you are saying, when the Hox genes form their proteins, these proteins can then regulate. But in the hierarchy, if you see, first there are their appearance of maternal defect, then gap genes, then pair rule, and then segment polarity. So within a gap, we have a pair rule. Within a pair rule, we have segment polarity. And if you see, Hox genes are determined by what segment polarity, what pair rule, and what gap gene the particular stripe is expressing. So as we have in the Hox genes. So what you were saying, like abdominal A will also must be true. Yes, I do not argue about that. But the order of appearance of these genes is this. I think if you face such a question, then it is Hox genes which are getting controlled. You should answer that. I'm not sure though, but this is what I also believe, that gap gene, pair rule, and segment polarity are the controller of homeotic genes, because and then homeotic genes can perform its function. For abdominal A, uh, we have to so this is what you were asking, I think. Yes, Ajay. Now can you tell me what confusion you had? Yes. Okay, let me see. Selector and regulators. Maybe I'm not able to find it. I have to I have to search Gilbert now. Posterior spiracle formation. Okay. Instead, if I show you something else on the Gilbert book, even I follow very little for Gilbert. I mostly rely on. Yeah, if you see this, the protein products of gap, pair rule, and segment polarity genes interact to regulate another class of genes, the homeotic selector, whose transcription determines the developmental fate of each segment. So this is where I also thought that regulation of homeotic selector is governed by the gap, pair rule, and segment polarity. And this is the exact picture that I put up from just the Gilbert and spiracle development. Let me see. Is this the region you were talking about? Homeotic abdominal B. Three protein coding genes are found in this complex in the bithorax, ultrabithorax, identity of the third thoracic segment, abdominal A and abdominal B. Yeah, the chromosome region containing the antennapedia is often referred to as homeotic complex. I think I have a different edition of the Gilbert, maybe. But as I was showing you earlier, there may be cases. Yes, yes. Ajay was saying something. Yes, yes. Please. Yes. Okay. Yes. Okay. 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 So that gap gene has a dual function, is what it means. So many proteins and many genes can have products. Yeah, as I was saying, many proteins and genes can have dual functions. You might, if you are curious enough, you might want to search moonlighting effect. You will find uh, very nice articles on that. A single protein can have two different functions. So EMX might be acting as a gap gene, which is laying down the expression of homeotic gene, which is the abdominal A. But here it is acting as a regulator gene, which is getting regulated by abdominal A. Yes, yes. So, but this is the basic notion that you should also believe in. Gap genes, pair rule, and segment polarity. Three of them will come together to determine the expression of Hox genes. Okay, that was a good discussion. Let's move on to the next question. Which of the following are distinct phenomena observed during fertilization in angiosperms? Which of the following are phenomenon during fertilization in angiosperms? Option A, double fertilization results in formation of sporophyte and endosperm. Option B, remaining accessory cells present in the embryo sac stay in the sac after fertilization. Option C, central cells contain two nuclei. When fertilized, produce a quadruploid endosperm. Option D, embryos developed by apomixis will lack a seed coat. Now, which of these are distinct phenomena for fertilization in endosperms? Let's take a minute to collect some answers. I'll wait for all the students to think about it. Then I'll reveal the correct answer. Ajay has answered option 1 and 4. Very good. Okay, it's time for me to reveal the correct answer. The first correct answer to this question: distinct phenomena observed during fertilization in angiosperms. Option 4: embryos developed by apomixis will lack a seed coat. And the second correct answer is option 1: double fertilization results in the formation of sporophyte and endosperm. Well done, Ajay. You have answered it very correctly. Very good. Okay, so the correct answer is option 1 and option 4. Let us have a look at both of these phenomena. So what we call as a double fertilization is very specific to angiosperms. What do we mean by double fertilization? So the male gamete has two nuclei. So the one of the nuclei, one of the sperms, fuses with the egg cell, singular egg cell, forming a diploid zygote, and the other sperm fuses with the polar nuclei to form a triploid endosperm. It's not quadruploid but a triploid endosperm. So the endosperm has 3N as its genotype and zygote has 2N. So this is the phenomena of double fertilization. And here is the structure of the ovule showing antipodals, synergids which guide the pollen tube, and sperms fusing with the egg forms the 2N zygote, and with the one of the sperm fuses with the endosperm to produce 3N of the endosperm. And the other option where we studied about apomixis. What is apomixis? Apomixis is a form of reproduction which does not involve fertilization. So it is an asexual form of reproduction, does not involve fertilization. If a seed is produced without fertilization, it is apomixis. And since the seed coat originates from cell layers surrounding the ovule, when we do not have fertilization, we do not have any ovule formation. Therefore, the seed will lack a seed coat, or the fruit will lack a seed coat. Therefore, apomixis leads to seeds without seed coats. And this is one of the ways in which apomixis can occur. Megaspore mother cell, or the female gametophyte, it usually undergoes meiosis, but if it is not undergoing any meiosis, it can bypass this and can directly lead to formation of a diploid structure, 2N egg cell, where the central cell will be 4N now. So even without, where the regular one would be central cell 2N and egg cell 1N, now because it is starting from a diploid megaspore mother cell, everything is doubled. So here we will have diploid egg cell but a quadruploid central cell. So this will develop into a fruit without any seed coat. Okay, it's clear to the students. Let's move on to the next question. Identify the incorrect statements regarding floral organ specification. Identify the incorrect statement regarding floral organ specification. Option A: in AP2 mutant plants, petals are made but sepals are absent. Option B: in AP3 and PI mutants, only sepals and carpels are present. Option C: in AG mutants, the innermost and outermost floral organs are sepals. Option D: the fourth class of floral organ identity genes is the sepalata. Okay, let's take some time and solve this question, you have to identify the incorrect statement regarding floral organ specification. So you have to remember the ABC floral gene organization, floral patterning. It's a very important concept, and this question is a revision for that concept. You have to find out which is the incorrect statement. Okay, Ajay is mentioning option 4. Okay, it's time I reveal the correct answer. The correct answer is, which of these is an incorrect statement regarding floral organ specification? The correct answer is option 1. In AP2 mutants, petals are made but sepals are absent. This is the correct answer. So let's have a look at the floral organization genes. We have many different classes of genes: A, B, C, D, and E. And the color coding here matches. So the blue ones, A, contains two genes, AP1 and AP2 belong to group A floral genes. So the first statement was incorrect because AP2 mutants will not only lack sepals but also petals. Because AP1, AP2 are involved. A group genes are involved in sepals and petals. In the A group mutant plants, petals are made but sepals are absent. So sepals will be absent, but petals will also be absent if we mutate the A group. And the other statement, in AP3PI mutants, what will happen if AP3PI are mutated? AP3PI mutated, no petals, no stamens. We'll only have sepals and carpels. Only sepals and carpels? True, it's correct. In AG mutants, innermost, outermost floral organs are sepal. In AG mutants, so innermost ones are the stamens and carpels. These are now uh, these are now uh, these are now sepals because of AP1 and SEP. So this will be a sepal without AG. Ovules without AG and stamens without AG will also be a sepal. So this is the correct statement. Without AG. So if there is no C, no red color, then this will become B plus E. B plus B will look like a sepal. So I think the innermost and outermost floral organs, the innermost and outermost sepals, this will look like sepals, and the innermost carpels will look like sepals when we have no AG. And the fourth class of floral organ identity genes is the sepalata. So these indigo ones. Okay, if you count, then these can be like the petal apetala genes from the first class, apetala 1 and apetala 2. Apetala 3 forms the second class, apetala 3 and PI. Third class formed by AG. So if you count, yeah, Ajay, you might be correct that it might not be the fourth class, but the fifth class of floral identity genes. I have, I might have to look into it. Sepalata, right? Sepalata is which group of? Okay, Sepalata is the E class. So E class, you can say fourth or fifth. It's controversial. But in AP2 mutant plants, so in AP2 mutant plants, would not have petals, and AP2 is a class A gene. So if you do not have AP1, AP2, then we will not have sepals. We will also not have petals. So that is the incorrect statement. In AP2 mutant plants, petals and sepals are absent. Both of them are absent. And I think option D is also the correct statement. Is it clear to you guys? Okay, let's move on to the next question. Embryonic mesoderm arises from dashed. Embryonic mesoderm arises from dashed hypoblast, primitive group, epiblast, embryonic ectoderm. Yes, Ajay, I know in blue book uh, plant biology is not given, but you can easily find it out on internet, or you can only refer to Subramaniam's lectures. They are very good. They'll help you understand, but you have to read it thoroughly. Sometimes you might confuse between genes and the floral arrangement. Sometimes some organs might form petals, might not. Sepals are everywhere because sepalata gene is everywhere. Now I was confused that whether sepalata is a fourth class gene or a fifth class gene. Because if you count it by alphabet, it is E class genes. I know that. A class are apetala and B are AP3 and PI. C are the AG genes and D are SHX. So E class genes are sepalata, but now whether they are called fourth class or fifth class, that is what I was confused. So you might be correct. Yeah, so you refer always to the recorded lectures of Subramaniam. Okay, for this question, pay your attention. Embryonic mesoderm arises from dashed hypoblast, primitive groove, epiblast, embryonic ectoderm. Which of these are a precursor to embryonic mesoderm? Hypoblast, primitive groove, epiblast, embryonic ectoderm. Okay, I see Ajay has answered epiblast, and it's time I reveal the correct answer. Well done, absolutely correct. Epiblast is the correct answer. Embryonic mesoderm arises from the epiblast. So here is a representative of a chicken embryo uh, development, which shows that the migration of these cells through the primitive group, migration of the epiblastic cells through the primitive groove forms a future endoderm. Cells moving over the sides of the primitive groove form mesoderm and endoderm. And the hypoblast is displaced by spreading endoderm. So the endoderm will be formed here and the mesoderm will comprise of epiblastic cells. Posterior epiblast cells change shape and thicken, forming the primitive streak. Cells migrate converging at the primitive streak to causing it to elongate. The primitive streak narrows and lengthens, forming the primitive groove, the chick blastopore. Cells migrate inward through the primitive groove and Hensen's node. Cells generated in Hensen node passing into the gastrula migrate anteriorly and form head structures and notochord. So these epiblast cells give rise to the mesoderm. Cells migrate through the primitive groove or Hensen's node and then uh, form these structures of mesoderm and the endoderm. Good, you were very correct in answering this question. Moving on to the next question. If a longer limb is advantageous in a given environment, the humerus may grow longer, but two smaller humeri joined together in tandem have never occurred due to what? So this is a specific example that was mentioned in the book in the lecture series. I hope you remember it. If a longer limb is advantageous in a given environment, why doesn't the why doesn't the humerus be constituted of two small humerus joining together, and why does the humerus grow longer? Two small humerus can also join together in tandem, but it never occurs. Why does it happen so? Option A, there are disadvantages in the process of natural selection. Option B, physical constraints. Option C, morphogenetic constraints. Option C, phylotypic constraints. Now, if you know it, you will be able to answer it in a jiffy, no issues. But if you do not know it, then it might be a problem. Why is a longer limb, if it is advantageous, it just grows longer, but not two smaller limbs joining together? Why doesn't it occur? I see Ajay has answered option 4. Okay. Okay, it's time for me to reveal the correct answer. The correct answer to this question is option C, morphogenetic constraints. Morphogenetic constraint is the principle via which the limb does not the limb does not join up with two small humerus, rather than it can grow longer evolutionarily. So if we learn the concept of morphogenetic constraints, it says that when organisms depart from their normal development, they do so in only a limited number of ways, or the body plan is fixed. The organisms which share ancestry will have the similar kind of body plan. If you see homology in the structures, there is homologous structures. We have one humerus, two radius ulna, then the phalanges, tarsals, and the phalanges. So this kind of body plan or the structure is repeated in all the animals which have common ancestry. So mammals, here we have the common ancestry as mammals. A human, horse, whale, bat, all are mammals. So they all have similar structure. One long bone, two bones here joined to the humerus, or it is known as the stylopod, zeugopod, and autopod, and then the digits. So the lengths might differ, but the plan is the same. We have two bones here, two bones in the whale. Therefore, if a longer limb is favorable in a given environment, humerus may become elongated, but one never sees two smaller humeri joined together in tandem. Although one could imagine the selective advantages that such an arrangement might give, this observation indicates a construction scheme that has certain rules. So only in a limited amount of space and the established rules can the evolution happen, can the growth happen. It cannot just produce like immediately or in a short period of time. It cannot produce drastic changes in structure, whereas we have two humerus joining each other and then two radius of line. So this is known as morphogenetic constraint. So it is one of the constraint that Professor Subramaniam also talked about, morphogenetic constraint, phyletic constraints. So phyletic constraint in the last week lecture series. Yes, yeah, phyletic constraint. Yes, yes, phyletic constraint says with a common ancestry, this has to go through this structure. Like even if that, like the example which we had, the notochord, the notochord present in the chordates, since now vertebrates are derived from the chordates, the notochord might not be present in the adult form, but it is transiently present in the embryonic form. So phyletic constraint tell us that the phylogenetic relationship, all the embryos in a similar phylogenetic relationship, closely related, has to pass through a certain stage. That is a phyletic constraint. Whether that stage is available in the adult or in the embryo, yeah, morphogenetic means morphogenetic means morphology will be preserved. But as in the phyletic constant, we saw that notochord is not present in the adult. So if notochord was still present in the adult, altered or somewhat, then we could have said it is a morphogenetic constant. Yes. With this, we come to the end of this session, bonus session where we solved around 11 to 12 questions, short questions, and I hope it was a good revision of everybody's concepts, and I hope it is helping both you guys and me to revise our concepts here. Yeah, let me just stop my presentation. Yes, yes, no issues, Ajay. At least you attended. I was very happy that we have at least some audience, because mostly even if students do not attend, or I still have to make the videos. So you might be hearing me like taking some time to gather some answers, and also people who will be watching these videos will need some time, so I wait for them. So so it's all that we have to do. We cannot force anybody to attend lectures, and also it's their benefit and all. So I was happy that you attended despite being busy. It's okay, and it's a revision class for everybody, since your exams are upcoming and all the modules would be revised in such a way so that you'll be able to remember some certain concepts and do well in the exam. Okay, the lecture recording will be available on the same YouTube channel, and the link will be provided. So you can, all the students can watch this, and all the best for your exam. This ends the Introduction to Developmental Biology TA course. You might never see me again. If ever it happens, I take this course again in the upcoming year, and you are still a student attending this course, then you can meet me. But mostly this is the point where we say goodbye. Bye bye. It was good taking a class with you, and I hope that you guys could at least receive, at the receiving end, you could receive something. Me as a student and a teacher, so it's quite a new experience for me to take classes for people, for professors, for students, where we have a uh, variety of audience. But I hope I could make things better and clear, and you could understand, you could benefit from the live TA session. That is all what we need. Okay, with that much, I'll end this call. Thank you for attending this course, and good luck for your exams. Thank you.